Good morning. I'm Nina Green. I'm the director for the Office for Technology Licensing. I'm going to just be very brief, and my remarks are really more logistical. The first is to remind you all that this session is being recorded, and if you have any questions, to please wait until the microphone is presented to you and you're holding the microphone, otherwise the sound will not be recorded. I just also want to thank everyone who helped me to organize this day: Nancy Santos Nicolau, Jen Carlin, Maria Spencer, Donna Barreto in particular worked really hard on putting this program together and making sure the speakers' presentations were up and running. And of course, all of my colleagues, Martin Sun, Jen Tsai, and particularly John Cosmopoulos, who really was the architect of the agenda for this day. And we really hoped, again, as to echo Peggy's remarks, that this would engage people in the process of what it actually means to develop drugs in an academic setting, and then the necessary next step to really work with industry in a way so that our early stage ideas can be translated and maximized and commercialized and ultimately get into the hands of the people who need the most, the patients. So again, thank you so much for the interest in this. We had a great number of people wanting to attend. I think it's going to be a really good day. So without any further ado, I will introduce Tony Coyle, who is the CSO and VP for the Center of Pfizer's Center for Therapeutic Innovation. And over to you, Tony.
Great. Thank you very much, Nina, and the organizers for the opportunity to share with you some of our thoughts about the evolving R&D ecosystem. And as we've thought over the last several months and years about how we can build a very different type of organization, one that leverages the understanding that exists in big pharma biotech around drug discovery and combining our expertise with the innovative science that exists primarily in leading academic medical centers. So over the next 45 minutes, an hour, what I'd like to talk to you about is this new R&D ecosystem. What have been the pressures to change from these insular, vertically integrated organizations that were very isolated to a large degree from the academic community? And how do we see the opportunity moving forward in partnership with institutions such as Tufts, really focused on translation? How do we identify new emerging science, understand the unmet medical needs for patients, and how do we identify these new opportunities that we can translate and accelerate that translation into meaningful studies in the clinic? I'll lead in then to some reflections on what we've established in Pfizer over the last year and a half, the CTI organization, an organization that's uniquely focused on identifying creative new ideas from academia and working in a very different type of partnership model. One that moves away from the traditional transactional interface with the academic community, but one that focuses on building teams, joint teams of clinicians, basic scientists, individuals who are interested in translation, and help infuse that understanding of what a drug needs to look like to be commercially viable. And of course, then to add a little bit of color, to give you a few examples over the last few months, working with many of our colleagues in the CTI network, to give you some examples about how we've seen opportunities that are scientifically exciting, where we've been able to work together to focus and build new therapeutics in idiopathic pulmonary fibrosis, targeting integrins with a collaborator in UCSF. Opportunities, we think, in cancer, targeting stem cells with an investigator from Children's, and with Beth Israel, a very exciting new approach to regulation of neoangiogenesis in the context of a variety of different tumor indications. So certainly during the next 45 minutes, if there's any questions that may come up during the presentation, please feel free to grab a microphone and we can try and make it a little bit more interactive, or certainly we can spend time at the end asking and answering any questions. I think there's probably very, very few people in the room who would not agree that the last 5-10 years has seen some incredible biomedical discoveries, whether that's even in the last few months, last year, understanding the importance of the microbiome, the interface of the microbiome with the host immune system, moving on from the tremendous effort spent on DNA cloning to understand the importance of epigenetic modifications. It really is quite disappointing, though, when we look and understand these great innovations, that translating those ideas into meaningful compounds driven by large pharma has been incredibly disappointing. This slide is a little bit out of date, but I think you can see just from the rapid decline and the slope of this curve that despite the change and the evolution in our understanding of the genetic and molecular basis for disease, these observations, largely in the academic community, are not being translated into new important therapeutics. So there's clearly a huge problem and a huge opportunity to address some of these challenges. How do we access innovation? How do we respect the intellectual property that comes from academia? How do we understand that, of course, the mission of academic centers is very different from that of Pfizer? But are there areas together where it's meaningful to combine our expertise in drug discovery together with the insights, the innovative ways that we can think about new discoveries that exist, again, largely in academic medical centers? You know, I think this slide is really quite telling as we look at the gap in the pharma pipeline. Looking at 5 large pharmaceutical companies plus Amgen. Looking at the peak sales, 2015, the new sales that are required to fill the pipeline and the number of products on an annual basis that is needed from Pfizer, GSK, and Merck to return value back to its shareholders. In the case of our organization, Pfizer needs somewhere between 8 and 10 new compounds every year to generate the revenue to support the huge infrastructure that exists within Pfizer. And I think if you draw the contrast between what's needed to sustain these large pharma organizations, and again, Pfizer is not alone in that respect. GSK requires 6 to 7, BMS 3 to 4 new molecular entities approved every year to support the pipeline and that return back to shareholders. Then, of course, you look at 2007, 2008, 2009, and 2010. In terms of the new molecular entities that have been approved, and I think you can see very clearly that striking contrast between the requirements and needs to return value to shareholders, produce new important medications to patients. We're very far from those 8 or 9 new molecular entities, with 0 new drugs approved from Pfizer in 2008, 2009, and again, not dissimilar from Big Pharma. So there is a huge problem within the industry. You know, and it's not just pharma that's challenged. Biotech is also in trouble as well, driven by capital and the drying up of capital that exists within smaller biotech organizations. The whole change in philosophy now that the valuation, of course, the interest of pharma around the opportunities coming from the biotech community is more shifted to positive clinical data at the end of phase 2 compared to 5-10 years ago when earlier stage innovations were very rapidly brought up by pharma. The requirement of what pharma would like to see from a biotech has really changed. The whole VC world, of course, again, it's a very different landscape than even 5 years ago. VC investments down, biotechs running out of cash, IPOs essentially have ground to a halt, and exits are via acquisitions. And certainly as a consequence, the implications of this changing landscape, fewer startups, valuations are down, and a tremendous conservative approach to VC investments in innovative new science. So pharma's challenged, VC money isn't there to support early stage biotechs, and that leaves the academic community in somewhat of a dilemma as how do we take those ideas from academia, working to understand what pharma needs to make those ideas into a format that would play and integrate into a pharma pipeline. So there has to be a way to think about different models to change and evolve the new R&D ecosystem. You know, I think this slide is also quite telling when we also look at where capital is allocated. If one looks at the clinical pipeline in biotech pharma, the pre-phase 2 pipeline, you'll see actually a greater dominance of clinical pipeline opportunities that exist in biotech. Close to 2/3 of pre-POC compounds are within the biotech space, and remarkably, only 1/3 sitting in big pharma. But then you draw the allocation of the pipeline, the clinical pipeline, to where cash is, and you'll see that big pharma has 90% plus of cash available compared to only 8% in the biotech community. You'll see the pharma spend, 98% of the dollars that are spent are being spent by pharma. It's not translating to new therapeutics. And again, 2% of the dollars that are in the discovery phase, pre-phase 2 space is allocated within the biotech community. So a total mismatch of capital and products, which is challenging to the biotech community and clearly, again, reflects the inability of big pharma in that traditional model to deliver new types of therapeutics. So it's really a perfect storm that threatens the traditional R&D model and has huge implications for how we think about working with academia. So clearly the big challenge in industry, the rising cost of drug development, a premier focused on innovation, and yet a challenge to access that new innovation. And productivity, as we've highlighted, a dramatic reduction in productivity, both in the biologic space and in small molecules. I think it's fair to say there's a tremendous pressure also in the pharma industry from investors, from shareholders, from patients who are really demanding different types of therapeutics. Pressures from the government and physicians, all again demanding new therapeutics that have a bigger impact in patients and in patients' lives. And then flicking over on the academic side, a tremendous focus on translational medicine. I think it's almost every week that we hear a new release from the NIH, from Francis Collins, stating the importance and the important role that academic medical centers can really contribute to the translational space. And yet that valley of death really exists within the academic community. How can we access and understand the various components that would allow individuals within the NIH, within academic centers, to understand drug development? And clearly we don't want to Pfizerize Tufts or anywhere else, but to understand the opportunity to take those great ideas, to understand the regulatory environment, to understand the implications from a manufacturing point of view, so we can translate new ideas into meaningful clinical studies. So a focus on translation, and those centers, those academic centers that have really invested in the translational medicine components, I believe will be the centers in the future. They'll be able to partner more effectively, be able to help investigators in the academic center move and take those ideas and increase value for the academic center, for the PI, for biotechs, and hopefully pharma afterwards. But we have to think of very different ways of partnering, very different ways that allows the individuals within the academic center, the basic scientists, the translational investigators, the clinicians, to leverage their expertise and again move away from this very transactional model that we've had in the past before. And I don't need to tell anybody in the room, of course, it's not just pharma that's challenged from a funding perspective. It's really very much the NIH-based funding with reduced government funding, despite the edicts around translation, really is the money and the knowledge there to allow academic medical centers on their own to translate those ideas. And it's some of these components that led us at Pfizer some time ago to think about building this very different type of ecosystem. One that's focused on a different type of partnership model, one by which we leverage the knowledge in the academic community, where we work with physician scientists who see patients, who understand the unmet medical need, so we can start to devise new types of therapeutics that are important therapeutics, that are not just me betters or second generation, third generation therapeutics that have minor improvements in the existing compounds that exist, but truly make an important difference and differentiated from, that we can demonstrate differentiation from existing therapeutics. And that's really the mission of CTI, the Centers of Therapeutic Innovation, is to partner with academic medical centers, again, to focus on translation and to really deliver on the promise of translating innovative discoveries from the bench to the clinic, demonstrating meaningful signals in the right patient population. I think it's fair to say those ideas and concepts of the blockbuster drug are probably ideas that were embedded, a decade ago. I think now, as we understand the opportunity to stratify patients, to understand that asthma, for example, isn't this monolithic disease, but based on our appreciation of the molecular factors that drive, be it lung inflammation, drive type 2 diabetes, or clearly in the oncology space, identifying the right patients with the right mutations so we can target the right patient population and deliver meaningful efficacy studies in rather small studies rather than these huge, large head-to-head studies demonstrating mortality endpoints, for example. So it's focused on identifying areas of unmet medical need, new therapeutics, and differentiation as being the key factors that we look for as we look for opportunities in the academic environment. This is some data published quite recently in a review in Nature Reviews that looks from the allocation of 252 drugs that were approved by the FDA from 1998 to 2007. I think it's really quite striking when you see where these ideas and new opportunities are coming from. And that's illustrated in the red bar, at least in the USA. More than 50% of drugs that have been approved have had their origin in academia or small biotechs. Again, it comes back to the questions about capital allocation. Where is the innovation coming from? It is not coming from large pharma. It's coming from academia. As we'll hear from our next speaker from Vertex, it's coming from smaller biotechs who can have the insight into new emerging science, understand how you can develop those new concepts into a therapeutic, and rapidly get drug approval in areas of high unmet clinical need. So there's a great opportunity as we think about big pharma, as we think about partnerships, to really address and tap into much of the innovation that exists within academic medical centers. So this is how we think about building this new ecosystem. It's been able to partner in a different way with academic medical centers where we can access basic research, access to patients. This becomes a huge challenge for large pharma. We think about precision medicine, we talk about the opportunity to stratify patients, and yet we are challenged to access tissues, tissue samples, to allow us to understand the opportunity for true patient segmentation. This again becomes profoundly important when we think about differentiation, is being able to understand the molecular fingerprint of diseases so that we can ask those important questions in the clinic in that right patient population. So understanding the unmet medical need, access to well-annotated clinical samples so we can understand both the genetics, the epigenetic components, and start to subdivide these large diseases into smaller populations that are meaningful and really focus on delivery of differentiation within a subpopulation of these broad diseases. Of course, what does pharma do? What does pharma do? Well, we understand drug discovery. We understand the commercial environment, we understand the regulatory landscape, and we know how to make drugs. We just don't know the right targets. We don't know the right patient population. So we have to bring and build these two different cultures together, which, is sometimes not easy. The philosophy of big pharma about, we can't share what we're doing because it's highly, highly secretive. Everybody's working on the same thing in big pharma, and the innovation there, it's a matter of who can get to the clinic faster rather than who's accessing the best innovation. So we do believe there's a significant overlap, and I fundamentally believe whether you sit in Tufts, at MGH, at Pfizer or AZ, there's one common goal that we share, and that is to develop new therapeutics that can treat diseases of high unmet medical need. So can we think of creative ways that brings together our expertise in drug discovery with these components of innovative science, of patients of opportunities of patient stratification and perform small, meaningful clinical studies that can address and confirm a mechanism about a particular target that excites us in the first place. And of course, then the implications are to focus on precision medicine, high impact in patients, novel targets, innovative discoveries that move rapidly to the clinic. You know, it was interesting seeing some of the data released in ASCO over the last couple of days from BMS, antibodies against PD-1. You know, this has been a molecule cloned close to 15, 18 years ago. It has taken us so long to go from that initial discovery into demonstrating very important readouts in the clinic in a variety of different tumors. If one could go back and think about that initial discovery of an inhibitory receptor expressed on T cells and B cells, and rather than spending 7 or 8 years performing a variety of different animal models, you know, curing mice with xenografts, looking at acceleration of floppy tails in EAE, if we believed in that concept that immune therapy in cancer has an opportunity, one could imagine that 18-year period from discovery to demonstrating meaningful impact in cancer, that really could have been a 3 or 4 year period. So it's thinking about acceleration. It's really focused on experimental medicine. Pharma can make compounds, we can demonstrate they're safe. So let's ask questions in the right patient population. And that means on the academic side is bringing teams together. So bringing teams together of basic scientists and teams of clinicians within that translational space. And I've been quite surprised over, you know, my last year and a half, how visiting UCSF, Rockefeller, Cornell, MGH, how you have some great scientists who don't discuss and partner with clinical investigators. And that becomes something very important from our perspective. It's really looking at the teams of people, building those teams within the academic community, teams of basic scientists and physicians, so that we can really enable and focus on translation. You know, while we speak about the budget in the NIH and the challenges of funding for academia, nevertheless, $31 billion is not a small amount of funding that's available to support academic research. As we think about the opportunities from a drug discovery perspective, to be able to tap into much of the NIH funding mechanisms, it becomes an opportunity then to co-share risk, projects where the government has funded research, that then with additional funds from pharma, we can then take some of the early observations, co-invest, co-work together, build this different model, and fund and take advantage of the taxpayer investments into basic science so we can translate those questions into patients afterwards. I think this slide that Francis has been speaking about more recently is also incredibly exciting. If you look from the early 90s with the unraveling and cataloging of the human genome, we now know many, many diseases, close to 4,000 diseases, that have a clear molecular and genetic basis. And that's been, I think, very exciting from the NIH perspective, from an academic perspective, to really understand the molecular basis that drives a number of important diseases. Despite that, in 2010, there was not a single approved drug where we know the molecular defect that causes the disease that biotech and pharma had delivered. Again, we'll see from some work from Vertex, of course, how that's starting to change. And it's great to see that in 2011, at least 1 or 2 new targeted therapeutics based on our understanding of the human genome have now been approved to treat disease. But you see that tremendous opportunity, 5,000 diseases, 250 with therapies that have been approved, tremendous opportunity based on understanding the genetic basis of the disease to develop new therapeutics in this space. So what's gone wrong in the past? Why have we lived in these 2 different worlds? You know, something as we've heard already. that changing culture, there is a big culture gap, and we have to find creative ways where we understand the needs of pharma, where we appreciate the ways that the academic community works, but find different ways that we can partner together. You know, in the past before, there's been these very large institutional grants. You know, Pfizer several years ago gave $60 million to Scripps. It's arguably and fair to say what has come out of that relationship with Scripps has been close to zero. And $60 million goes a hell of a long way when you think about funding research. So large grants, but generally with a low return on investment. You know, we've all had the opportunities to be part of licensing and sponsoring research agreements, but relatively short frame in the goals of those focused on very specific questions without building that longer-term relationship, without adding more value. You know, there's certainly the appreciation, I think, coming back to the culture gap, you know, that folks within the academic community, within the pharma community, you know, may not be quite as smart as your average academic scientist, and the perception in pharma that folks in the academic community don't have the same rigor as one would need to see as we think about drug discovery. And then this whole collision of priorities. You know, pharma's tremendous desire in the past to control product development, intellectual property, being very conservative how we think about publications, and of course the concern that working with academia, you know, when there's a grant from the NIH, you know, there's certainly a desire, which I fully appreciate, to explore a little bit outside what the initial scope of the funded research would be. So these are the cultural aspects that I think we have to think very seriously about, and how can we ensure that our goals are aligned, that we all believe we're moving in the same direction to ask the same question afterwards. But I think critically to move away from this transactional approach is going to be essential if we're going to work effectively with academia and with pharma together. So it's all about delivering value to stakeholders, and that was embedded in many of the concepts that I'll start to share with you as we thought about the opportunities within the constraints and confines and opportunities embedded therein within CTI. So we thought we needed to work together in a different structure, deploying people and technology, building different types of teams, building a different type of partnership model that enables speed and flexibility, focused on long-term relationships with academic centers, building trust and working through the tech transfer offices. And I'll make a quick plug for Tufts. One of the most forward institutions that we've had the opportunity to partner with has been the Tufts organization, the medical school, and Tufts University. The goal is, of course, then is to focus on delivering meaningful opportunities in the clinic, and hence bringing together these teams and working on teams whose goals are aligned, that we understand we're working together to ask this question and focusing on delivery of that question afterwards. And again, the projects that we've been excited about are those projects where the science is exciting, where the investigator who may have spent many years asking questions in NOD mice or performing xenograph models, which are important, but we'll be able to take those individuals, partner with clinicians, and then work together to ask questions in the clinic afterwards. So this led us to how we felt about this new ecosystem model and building the group that we've referred to as CTI. So it's focused on strengthening the scientific core in Pfizer, certainly from our perspective, building a new entrepreneurial group to provide additional opportunities to focus and partner with leading individuals who have expertise in targets, in mechanisms, and in pathways. It's focused on externalization. Let's move away from our large facilities in Groton and Kalamazoo, in St. Louis and Pearl River and other places that I've yet to go and visit. So thinking about partnering with leading academic centers in Boston, in San Francisco, in New York, in San Diego, centers where there's a tremendous entrepreneurial culture, where we can partner with many different academic medical centers within a framework that's pre-negotiated, so we can access literally hundreds of thousands of scientist agreements, and do this in a pre-arranged structure where we have predetermined and pre-negotiated milestones and royalties. And as Nancy and Nina know very well, we don't need to sign CDAs or MTAs. These are all covered under the agreement. So we don't spend 9 months negotiating, that we can see great science together, enter into that partnership, work through these steering committees that are comprised of members from Tufts Medical, for example, with colleagues from CTI, and together we identify the best opportunities, the best individuals that we can translate to the clinic. And of course, again, it's all about differentiation. And maybe one important example of this is in the rheumatoid arthritis space. There's probably 12 biologics approved in the RA space, 6, 7 anti-TNFs, a number of small molecules, including a new small molecule from Pfizer. They'll be approved by the end of the year. So while there is still an unmet medical need in rheumatoid arthritis, the opportunity to develop a new biologic in that space and to show differentiation is close to impossible. So again, it's coming back to the patients, it's understanding again the unmet medical need, and it's an appreciation of the commercial reality. There is no opportunity, certainly in our belief, to develop a new therapeutic in rheumatoid arthritis unless you can identify that subpopulation of patients with RA who will be TNF refractory patients. Then this opens up a whole new possibility to target a subpopulation of patients and demonstrate meaningful efficacy and true differentiation. So positioning ourselves for differentiation, so it's collaborating now with PIs who see patients, who we can think about patient subsegmentation, focus on precision medicine, and demonstrate in the clinic meaningful differentiation. And of course, it's not just pharma, but one thinks about the reimbursement landscape. Unless we can demonstrate true differentiation, for these expensive protein therapeutics, patients will not be reimbursed if we can't differentiate these new innovative therapeutics from the existing standard of care. So while we have to focus on innovative new science, I think we have to be very, very mindful about the landscape that we live in, the reimbursement landscape, and differentiation, unmet medical needs should really be forefront as we combine innovative new science with how we can make a difference to patients and understanding the regulatory and the reimbursement environment. So this is how we think about CTI, and this is our vision as we set up this group some 18 months ago. The unique focus of CTI is to translate novel discoveries from academic centers into early stage proof of mechanism in the clinic. So we're not trying to cure diseases in CTI, but we want to ask those meaningful questions in patients to confirm the mechanism that excited us about the targets in the first place. Pfizer is a huge organization and somewhat bureaucratic. I hope there's no... We are being recorded, aren't we, Nancy? So we felt it was important, nevertheless, to build these small teams that are biotech-like in nature, where we co-locate people with the sole focus of rapid acceleration of new ideas to the clinic. We understand what we want to do is to build these teams that are totally complementary in expertise. We want to bring our expertise to the table and partner with academic medical centers. And it's capital light, it's efficient, it's a flexible funding process, and a very close collaboration where, again, our goals are aligned. And if I reflect on the last year, that's probably one of the most important learnings, is to identify people who share that same common aspiration to translate a new idea. And it certainly doesn't work for everybody. Whether it's understanding more about, genetics from Drosophila experiments, which are profoundly important. The sole reason we established CTI was to work with like-minded individuals who want to see basic science translated. So what have our been successful factors? Why do we believe that CTI does represent a very different way to work with academic communities? As I highlighted, to build these clusters and co-locate people I think this is profoundly important, and for folks who have been to some of our CTIs, again, we're embedded in the middle of academic medical centers. I have a UCSF badge, and when I can walk into anywhere on the UCSF campus, and the folks in UCSF who are partnered with CTI California have total free access to our facility. So again, it's building that transparent exchange and combining the best of pharma and biotech. It's thinking about, again, translation. We've mentioned Francis, the NIH, working with academic centers where we can build those teams together. It's sharing risk. Many institutions have a track record for financial support for drug discovery. We're working very closely with the Stanford Burnham in California. A great understanding of what it takes and the concepts of high throughput screening. Strong life science innovation community. We started off CTI with UCSF, a tremendous entrepreneurial community that helped us think about the challenge and how we partner differently with academic centers. And having this entrepreneurial spirit and ingenuity. And it's taking risks. That's a flip side of innovation. It's taking risks with new ideas. And that's really the context of CTI. If it's going to be a new idea, we're going to know a lot less about that concept. But if we work together, let's make sure we understand what we don't know, and let's partner together to address the questions that we don't fully appreciate. So the CTI model, we wanted to keep the whole governance light. We felt that was very important. We're a semi-autonomous organization. And we have here our three main geographic hubs. We're adjacent to the Gladstone Institute in Mission Bay in San Francisco. In New York, we're on 29th and 1st Street in the Alexandria Center for Life Sciences, right next to NYU. And our CTI Boston hub is over in the CLSC building, over in the Longwood Medical Community. Again, we felt it was important to get out of a Pfizer facility, and individuals who partner with us have an access card and come and spend as much time in these facilities as they so desire. So a very different way of working with the academic institutions. And this is our network to date. This is what we've established over the last 12 months. Again, the first CTI in California partnered with UCSF. The second CTI in New York City with 8 different institutions, with NYU, Cornell, Rockefeller, Sloan Kettering, Mount Sinai, Columbia, etc. And then, of course, CTI headquarters based in Boston, where I'm located, in Longwood, in partnership with Tufts University, Tufts Medical Center, Beth Israel, MGH, Harvard. So 20 institutions that are part of our network. We have a small group actually housed in a Pfizer facility in La Jolla in partnership with UCSD and Stanford Burnham. It's quite remarkable looking back that all these 20 institutions have signed exactly the same agreements. And I can assure you there were some, interesting moments of last-minute diplomacy, trying to hold NYU and Cornell and Rockefeller into the same agreement. But I think it reflects how we thought about how we need to build a different agreement that creates value for our organization, clearly, but creates value for the academic institution. And as I highlighted, gives us access to literally tens of thousands of physicians, basic scientists, all of whom share that same common goal focused on translation. Again, coming back to what we think makes a successful collaboration. The steering committees that we have in place in all of the different schools, they're a bit variable. The culture that exists on the MGH campus is very different from the culture that exists in NYU or in Rockefeller. And again, I highlighted the terrific interface with the tech transfer offices, certainly in Tufts Medical and Tufts University. And it's really having those key figures on campus that allows us to access the right individuals who may have this new emerging idea that's not yet published, but could partner with us to help make that idea ready for prime time. So it's a steering committee, it's having those advocates on campus who are believers in translation and partnership, and is having a sophisticated tech transfer office that understands the value and the long-term value of the type of relationship that this could bring. The steering committee, as I mentioned, is really important for us. These are joint decision steering committees that help us identify not just great science, but help us identify the right individuals, that these would be good partners to work with. Making sure that we're aligned in terms of the goals and doing this in a rapid manner, making decisions quickly, low on bureaucracy, high on self-autonomy, and again, working together quickly. And again, working with engaged physicians who have that true desire to see a preclinical concept translated into a meaningful POM. And it's been very important for us at the onset of selection is to find the basic scientist who has to partner with his or her clinical buddy so we can start to think about, again, precision medicine and start to understand even very early, how would that first in-patient study look like? What would be a meaningful signal to see in patients to give us the confidence to further invest money to take that end of phase 1 study to a phase 2 study afterwards? And again, it's working side by side. It's getting those postdocs working in our facility, coming and going back and forth, accessing our technology in terms of our phage display capabilities, accessing our Pharm-Sci organization, and this truly makes a difference for those individuals who want to partner with us. So we've had the opportunity to review over 300 proposals over the last 12 months across a variety of different therapeutic areas. And we don't care in CTI whether it's in inflammation, oncology, in infectious disease, neuroscience. If it's a good idea, if it's the right team, can we translate that into the clinic? Then let's move forward with that. What we've tried to put in place is a 2-step process by which we see those great new ideas early. Then we build a team. We build a team where we put a Pfizer clinician, where we put our individuals from toxicology, from Pharm-Sci, our assay development people, so we can take that idea and make it look like a drug discovery research plan with a demonstration and articulation of what a meaningful PM would be in the clinic. And to date, we've funded 23 programs in really 4 major areas: rare diseases, which we think there's a huge opportunity in, oncology, inflammation, and cardiovascular disease. So I have about 10 minutes left. I'll just run through some of these programs quite quickly, just to give you some reflection of what we have felt have been interesting opportunities and programs and the types of people we've been collaborating with. So in Boston with Marcus Frank, targeting cancer stem cells, really based on some very exciting data that Marcus demonstrated some time ago, that this ABCB5 positive population, it's a ATP cassette family member transporter, is really a bona fide cancer stem cell antigen. Marcus partnered with Steve Hoddy, who runs the Dana-Farber Melanoma Center, and they demonstrated using several hundred patient samples that ABCB5 is expressed in malignant melanoma and in malignant melanoma with metastatic capability and is not expressed in benign tumors. Marcus also demonstrated that this is a true cancer stem cell antigen in that when Steve and Marcus took ABCB5 positive cells from patients with malignant disease and transferred those back to mice, is the ABCB5 positive population that results in primary tumor formation and results in the formation of secondary tumors afterwards, demonstrating that this is a true cancer stem cell antigen. Marcus has made antibody against the extracellular loop and the third domain of ABCB5 and had demonstrated in his science paper that antibody inhibits tumor formation through an antibody-dependent cell cytotoxicity mechanism. But of course, his antibody is far from being a drug. So now working with Marcus, we've taken his initial observations, we've taken his mouse anti-human antibody, we're affinity maturing, humanizing, modifying the effector function, and we believe with Marcus and with Steve, we could be in the clinic in around about 18 months in patients with stage 3 melanoma. That's a tremendous change, again, using our PD-1 example. Again, reducing that 18 years down to what could be a 3-year period to ask a question in melanoma where there's a huge unmet medical need within those stage 3-4 patients. Likewise, over in San Francisco, we're working with Hal Chapman, who's the head of the Interstitial Lung Disease Clinic. And Hal has demonstrated an important mechanism to regulate the fibrotic components through an alpha integrin-dependent E-cadherin interaction. And again, what excited us about working with Hal, he runs the Interstitial Lung Disease Clinic in UCSF, and he has literally access to hundreds of patient samples, sees 250 new patients with IPF per year. This is an area where the mortality in IPF is not dissimilar from many types of cancers, and yet we don't understand how to understand the heterogeneity of this disease. So working with Hal now, we're trying to understand biomarkers that will predict individuals who have a rapid loss of lung function. And through the work we've devised with Hal, understanding a mechanism to regulate TGF-beta production to maintain the normal immunosuppressive components of TGF-beta, which is clearly essential in tissue homeostasis, but to modify the pro-inflammatory roles of TGF-beta in the context of EMT. I'll spare you some of the data just in terms of the time that we have, but I think just to highlight what we can do with Hal, he demonstrated through an elegant series of mouse experiments the critical role of alpha-3 activation in TGF-beta mediated signaling, but equally important, could demonstrate in the lungs of patients with IPF that signaling through this novel integrin E-cadherin TGF-beta R1 tripartite complex, there's a non-canonical activation resulting in beta-catenin phosphorylation in alpha-smooth muscle positive cells, with the formation of these beta-catenin SMAD2 complexes. This, for us, would constitute a positive proof of mechanism in IPF patients. Again, we're not trying to cure disease with Hal. We want to make a new therapeutic that prevents an interaction between alpha-3 beta-1 E-cadherin. Then ask a question in patients with IPF and demonstrate reduced phosphorylation of beta-catenin to give us the confidence then to move on into a large trial in patients with IPF. And finally, working with Hal Dvorak, I'm sure many of you may know Hal, clearly the godfather of angiogenesis and the discoverer of VEGF many years back. And what Hal has been investigating over the last 5 years are novel ways to target neoangiogenesis in the context of tumor formation. And a very elegant series of experiments looking at the formation of new blood vessels that are VEGF-driven, early blood vessels, those mid-stage vessels driven by VEGF, and late-stage vessels induced by overexpression of VEGF. What became important in this context as we spoke to Hal about the opportunity here was, again, thinking about differentiation. While these early-stage vessels are uniquely dependent on VEGF, once there's established angiogenesis with this network of new vessels in late angiogenesis, VEGF inhibition is no longer effective. So one can start to think about creative ways that are truly differentiated from the standard antibody therapeutics targeting VEGF through antibodies, traps, and otherwise. We've identified a surface molecule that's expressed on these late-stage vessels, TM4SF1, I'm working now with Hal. We're making antibodies, the existing antibody that's been generated from Hal. We're humanizing. We're using our proprietary bioconjugation technology to make a TM4SF1 antibody drug conjugate for patients with a variety of different tumors. And to date already, even after several months of working with Hal, we've confirmed some of the expression that TM4SF1 is expressed in the colon, kidney, in breast cancer, and making an antibody drug conjugate, we can demonstrate that Hal's initial antibody binds to the cell surface of late-stage blood vessels, in this case shown as proliferating HUVECs, is rapidly internalized to endosomes, and using our technology with antibody drug conjugate bioconjugation can demonstrate that we can very effectively kill these blood vessels in a manner that VEGF inhibition doesn't touch. So I hope to give you some idea of how we can take, again, a great idea with a leading investigator such as Hal Dvorak, use our technology to make a very different type of therapeutic. That again, we can take that idea and move to the clinic quickly. So concluding remarks, CTI and academia, we believe it's a very powerful combination. It's building a long-term relationship through a truly collaborative partnership. We're not focused on any different diseases, so we can really focus on great new science and translate that, breaking some of these traditional boundaries that I think impede pharma. When the neuroscientists have to build a neuroscientist portfolio, we in CTI are interested in good ideas, irrespective of the therapeutic area, that we can translate in a seamless manner by partnering with basic scientists and academic clinicians to translate new discoveries in a seamless manner from discovery into the clinic. Of course, from our advantage, we can move away from our large insular organizations, but truly build these small groups embedded in places like the Longwood community. Enabling translational research. We understand the types of toxicology experiments that we need to do to file an IND. We can make GMP material to enable that phase 1 study. So we can really bridge the basic science, the clinical execution ability within the academic centers, and leverage, again, our understanding of drug discovery. And of course, again, incentivized to take us all in the right direction. Incentivized to ask questions at the end of phase 1 and the end of phase 2. So CTI and Pfizer, we're still a relatively small group. We're a group of about 100 people out of an organization of 10,000 in R&D. Nevertheless, there's tremendous focus on how we can drive innovation in a focused manner, truly sustainable, winning in Pfizer through superior quality, with innovation with our academic partners, and most importantly, again, whether it's in Pfizer or in the academic community, really focusing on unmet medical needs and produce new medicines, new vaccines that matter most to patients and make a huge difference in patients' lives. So with that, I'd just like to acknowledge the folks who I've been working with over the last year or so. Some very talented lawyers that crafted an agreement that 20 academic institutions were able to sign. Certainly Justin and Louisa. Jose Carlos Gutierrez Ramos, who is the head of biotherapeutics within Pfizer. Torben, who runs project management. This has been very important for us as well, to have project managers working side by side, our scientists and postdocs, investigators in the academic community. And finally, just to mention a brief word about the individual who helped me set up CTI, Alex Fain, who very sadly passed away a few weeks ago. So with that, thank you very, very much for your attention. I'm more than happy to answer any questions. Thank you. So currently to date, we have 23 teams that we've established across the entire network in 4 main therapeutic areas: rare diseases, oncology, inflammation, and CV Med. So we're funding that's around about 7% of the opportunities we've seen that we've funded. So we think it's not too different from NIH-based R01 funding and the percentages funding afterwards. And we fund those projects to the tune of about $350K per year. Again, very much in line with the NIH funding rates as well.
Thank you very much for your perspective and this very interesting talk. It sounds as if you have reached major milestones, and the question is, so what's left to do?
This is just the beginning, my friend.
So just to widen this concept and expand it?
I think that there's several, horizons that we saw that will determine our success. The first was to, build labs, identify the best science, build this portfolio of 23 projects. I'm very proud when we look at those projects that are currently part of the network. I think only 1-2% of those opportunities even overlapped with the internal Pfizer pipeline. So we're seeing something that's very different. What we're seeing over the next 2 years are how many of those 23 opportunities can we identify candidates that are ready for regulatory talks, and how many of those would move into the clinic and demonstrate a meaningful signal afterwards. So we're moving from that initial phase of identifying the opportunity, starting to develop those candidates, and then of course asking the translational question in the clinic. What we would like to see as we move into 2013 is to build a much larger portfolio, one that's around about 35, 40 projects, to diversify from large molecules and think in very creative ways. How can we use the same principle, the same network, to understand the opportunity with the small molecules? We understand there's a lot of target space we're not even looking at. So really two things for expansion. One, it's the modality, the geographic scope, and then thinking about the clinical execution. More creative ways that we can perform clinical trials.
May I add another suggestion to that? And this is, what I see is the transition point is at the translational research. Mm-hmm. But research in academia starts much earlier. And this is where I see a tremendous funding gap with regard to lack of seed funding in order to mature early ideas to the level where they reach noticeable attention and where one can actually estimate whether there might be a chance for a new therapeutic approach or not. So this seed funding, what is not taken over by VCs, that is not taken over by angels because they don't, in many cases, understand what the business is. And this is where the budget of academic institutions face an enormous difficulty, this incubator function.
Just a comment, and I totally agree with you. I think that's one thing that we've learned. While we've seen some great ideas, we've been focused in CTI version 1.0, if you will, on those ideas that are ready to move forward and translate. We've seen some fantastic stuff that we felt was just a little bit early. As we move into 2013, that is exactly what we need to do, is to set money aside. Small amounts of seed funding, maybe funds for 6 months, 9 months. If you can do X, Y, and Z, this then would be ready for the full CTI investment afterwards. But I think you're absolutely right, and that's something that we need to implement next year. Totally agree with you.
I think in the interest of time, thank you so much.
Great. Thank you.